Good morning. Welcome, everyone, to day 2 of our workshop. Yesterday was absolutely fantastic, and not only did I learn a lot, but I think we managed to break many language barriers, as it were, in trying to understand the concepts and models that people across disciplines use when thinking about the kinds of inference and the sort of pitfalls in many of the methodologies that we're using, the limitations across fields. So it is my great pleasure today to introduce our Dean of Faculty of the Faculty of Arts and Sciences, Professor Tamar Gendler, who is an exemplar of interdisciplinary scholarship and whose support, in her own work, she has worked across multiple fields, philosophy, psychology, cognitive neuroscience, and is very committed as an administrator in the university. She has been really heralding sort of intellectual crossing of boundaries and many initiatives and programs to really facilitate conversations. As you all know, universities, though we are all in with various expertise that often speaks to very similar sets of problems and challenges that we work on, we don't get to talk as much. So with the support of the Franke Program in Science and the Humanities that Yale has very enthusiastically not just supported, but encouraged faculty to attend and be involved, Tamar has, as I said, in her own scholarship and beyond, really been a steward for cross-disciplinary work. So it is my absolute delight to invite her to say a few words. Tamar, thank you so much for coming.
Thank you so much, Priya. Thank you all, and I'm sorry I wasn't here to greet you yesterday morning. These are, as you know, challenging times for universities. I was in New York helping some of our colleagues and supporters understand how hard we're trying. But though these are trying times for universities, this conference is an example of what makes universities truly distinct and what it is about the imaginative enterprise of creating an institutional structure that is simultaneously engaged in the preservation, the creation, and the transmission of knowledge. And in my mind, this conference epitomizes what is great about universities. It is asking a deep and intellectual question that is simultaneously of theoretical and practical significance. It is distinct, I think, to be able to have a series of events that simultaneously engage some of the deepest and most abstract philosophical work, really about the nature of understanding in its most fundamental sense, and that simultaneously brings in very real, practical ways, a set of empirical disciplines. It's rare to find a topic that is not at the periphery of one field and the center of another, but at the center of every field of which it is a part. And it is rare to find a subject that, though centrally part of each field and common enough to merit joint discussion, is differentially illuminated by different methodologies. So that's some theoretical reasons to think this series of events is doing what a university can do. But I also want to point out, because I have a second hat, I'm not just responsible for intellectual matters, though I take great joy in reading the academic work of my colleagues regularly. The second thing I want to emphasize is that this series of events, of which all of you are a part, took advantage of structural features of the university, of structural features of the ecosystem of which we are all a part, and to the extent that the group I am addressing is a set of stewards of the intellect at their own universities, I want to point out to you some of the practical features of this event. This project was carried out over years. It used multiple modalities. It took advantage of the fact that people can drop in and out and nonetheless be part of a loosely knit social network. For each of the events that it carried out, it had it both in real time and available for subsequent observation. It brought unexpected groups of people into interaction with one another. It took advantage of the funding environment in which we find ourselves. The Templeton Foundation is deeply interested in interdisciplinary issues, in fundamental questions. It supplemented those resources with an entity on campus, the Frankie, that had already established a mailing list and a brand that allowed these events to be legible. And it involved a wide range of faculty on and off campus. Priya did the very smart thing of very early alerting this to me. And so when I went around and met with supporters of the university, this was one of the things I talked about. Every single thing that Priya did with regard to this event is something that every single one of you can do at your home institution. Think about what are the networks that have already given us a pre-established list of individuals with interest in a large range of questions. Who are the nodes at your university that if you tell them about this, they will tell the world about this? This is not only in your self-interest as scholars. The university in the world is an entity that is at risk. It is being challenged for reasons, some appropriate, some inappropriate. But the more we focus and celebrate core mission, the more likely it is that universities will remain the entities that they have long been. So that's where I was yesterday. I was in Defend the University space, and I was writing this on the train, and so I wrote you a little Defend the University. But I also want to do a couple of specific reflections on the topic that this group has been thinking about. And then I'm going to just close by reading a poem because this doesn't have anybody from the English department, and so I thought I'd give you a little bit of literature at the end. So on the front page of the web page, which I assume is the cover page of the proposal that you sent to Templeton, it certainly feels like the cover page of a proposal that one would send to Templeton, is the incredibly resonant quote from Hume that no amount of observation of white swans can allow the inference that all swans are white, but the observation of a single black swan is sufficient to refute that conclusion. And I don't get to do a lot of philosophy. I don't get to do a lot of reflecting on texts. So it was a total pleasure just to live in this quotation and its depth. And I just want to give voice to three thoughts that occurred to me. One, I had not realized till I started thinking about this quote that one way to describe what almost every academic does is to figure out which features of the world that seem shallow are actually deep, and which features of the world that seem deep are actually shallow. That is, for example, the project of our Department of Women's, Gender, and Sexuality Studies. Which aspects of the world that seem universal may be particular, which aspects of the world that seem particular may actually have deep foundations, as I suspect we will hear in our next talk. But over and over and over again, including in the work that we do in engineering, which ways in which this substance feels brittle have to do with deep features of its structure, which are merely manifestations of the idiosyncratic way in which we find it. So that's just thought one. Hume tells us a way to think about the questions that we ask is to think about where we are mistaking surface for depth, where we are mistaking depth for surface. Ultimately, a Platonic question about our capacity to understand the nature of reality. 2, if Hume had lived in Australia, it wouldn't have occurred to him that all swans were white. In Australia, lots of swans are black. It is contingent what it is that we take to be ordinary and what it is that we take to be unusual. That reminder is crucial for all of our investigation. That is not a political statement. It is an epistemic fact about the world that all of us are only partially exposed to elements of it, and on that basis, we draw conclusions. Note that as we build artificial analogs to our own intelligence, they too don't live in Australia. That is to say, they too are getting exposed only to that which is electronically represented. And just like Hume, who was very, very smart, they might fail to recognize how idiosyncratic the part of the world to which they have been exposed is. Third point, how do we gather information about the world? One of the questions that this assembly is thinking about is the entire set of methodological worries that arise. To what extent do we seek information? To what extent does seeking information distort the information that we're able to get? How do we become active perceivers? I spend a lot of time, I oversee all of the tenure and promotion cases that come before us. I see the methodologies that ethnography brings us, which says sit back, don't theorize, and you will see things more truly. I see the methodology that's brought by areas of inquiry that say if you do not have a theoretical framework, you will mistake what is important for what is unimportant. Both those things are true. And the methodological diversity of this event seems to me to capture that in profound ways. All right, those are 3 thoughts on Hume. Let me close with a wonderful poem by Mary Oliver. This is not a hard, deep poem to understand. This is just a beautiful celebration of our experience as humans on Earth. The swan. Did you see it too? Drifting all night on the Black River? Did you see it in the morning, rising into the silvery air, an armful of white blossoms, a perfect commotion of silk and linen as it leaned into the bondage of its wings? A snowbank, a bank of lilies, biting the air with its black beak. Did you hear it fluting and whistling a shrill, dark music like the rain pelting the trees, like a waterfall knifing down the black ledges? And did you see it finally, just under the clouds? A white cross streaming across the sky, its feet like black leaves, its wings like the stretching light of the river. And did you feel it in your heart, how it pertained to everything? And have you finally figured out what beauty is for? And have you changed your life? Thank you so much for inviting me and for your assembly.
Thanks, Tamar, for that very, very lovely welcome, and thanks to all the attendees who are here. So we are opening this morning's session with the first talk. Eduardo, take it away.
Thank you. Can you hear me there? Yes. Well, good morning, everyone. I want to stay on time. Thank you, Priya, for inviting me. I'll have a few words to really share with you all why is it that I'm here. Thanks, Tamar. I'll have some comments about what you were referring to before. You have the title there. We're going to move to look into some different type of causation. Causation, the way we examine it in biological anthropology. And I will say more about that in a minute. What I want to do first is get the clicker to work. There you go. I think it is important. I put together this slide, which I don't usually show, to give you some background on my own background, right? I'm not a philosopher, that much you've learned from yesterday. I'm not a computer scientist. I'm not a statistician. I'm a biologist by training, and with a focus on behavioral sciences. And that has got me to move across disciplines. And I really think that maybe it's helped me to really appreciate and embrace the opportunity that this workshop is bringing to all of us. My very first research paper was working in a lab that studies the neurobiology of learning and memory. We were using some short crabs that we were collecting from the beaches in Buenos Aires, bringing them to the lab, giving them electric shocks to see if they could learn or not. They don't. But then I went on to the PhD to work with some experimental psychologists. So I went from neurobiology to experimental psychology, tackling some of the same questions that I will be presenting to you, but in a controlled setting of a lab at the California Primate Center with my mentors there. So I'm going to come back to this because I think it's one of the things that I would like to share broadly with my colleagues in biological anthropology. And I was discussing with one of you over breakfast. I think there's a lot to be said about the benefit of taking the leap and cutting across disciplines as you develop your own trajectory. The compass for the talk. Some of you still remember what a compass is. Some of you only know about GPS. So what I want to do is give you a little bit of a few minutes of why I'm talking here today. Then I'm going to make a few comments on causality in the biological sciences, a little bit of an introduction to how we do that in biological anthropology with some very general examples on questions and the type of evidence we collect. And then the core of the 30-minute presentation will be on some examples from my own research on pair bonding, monogamy, and fatherhood. I really plan to be done in 30 minutes so that we can have full 15 minutes of questions and answers because I have enjoyed those so much, as well as our times together over breakfast, tea, and lunch. Why am I here? So sometime, I think it was in 2018, I had my first teaching relief and I went on leave. And I used that semester, so that is 5 years ago, to really start pondering and reflecting. I want to know what I know, right? Sometimes you feel like I only know that I don't know anything, but I wanted to know what I know. And I did. I spent a lot of time on that semester. I put together a course on scientific thinking and reasoning. And one of the books that I went back to, I knew this book, I don't know how many of you know it, it's from two British colleagues, Stop Working and Start Thinking. And I think that cartoon says so much of the challenge we face on a daily basis. We can seldom find time to stop, ponder, reflect. I mean, just the pressures to get money, we don't even know what we need the money for, but we just need to get more money. And so in 2019, Dean Gendler launched a program that was absolutely brilliant, I think. It continues to run, which was the Teaching Relief for Learning. We were invited to come up with an idea. We needed to convince them that we could make good use of that time to take courses instead of teaching courses. Go back to the classroom. And that was a time when I was starting to say, listen, we're not reflecting on what we have learned in a causal inference sense within my own research. I mean, I have been doing my research now for 25 years. So I knew that some of the social sciences were developing all kinds of methodological tools to get at causality. And so I literally enrolled in a research methods course in causal inference, taught in political sciences, and then I took an undergraduate course, one of our survey, introductory courses in stats. And I did both, and I completed my homework, and I passed. And it was fantastic. It was fantastic because I realized that, oh my, do we need to bring some of those tools into biological anthropology? I continued working there. There are all kinds of developments that I'll be happy to share with you because like Tamar said, maybe some of these ideas you'll bring to your institutions. But it was only a few months ago, as I was developing a new course on causal inference, that I just went onto the Yale website and typed causal inference. And I will admit here right now that I only learned about the causal inference project that we have at our institution maybe 3-4 months ago. And yes, the more you work, the luckier you get. I emailed Priya and I said, I want to know more about this. And that got us talking, and I jumped on the wagon of what Priya has been doing, and so that's why I'm here. Only because I was seeking information, I was looking for ways in which I could expand my intellectual interest and prepare myself better for doing my research. So that's my history of why I'm here. A little bit of the history. I mean, biology has a long history of thinking and contributing to the development of statistics. I mean, this is just two graphs from a wonderful book that most of you probably know, The Book of Why by Judea Pearl. It's Sir Francis Galton towards the end of the 19th century, just developing ideas for explaining the inheritance of height in humans. I mean, at the same time developing the central limit theorem. And then Sewell Wright, this is a figure 1 from a paper in PNAS. My understanding is that historians of science consider this the first path diagram. He was trying to understand what causes color code in the guinea pigs that he was studying. So late 19th century, early 20th century. Move forward and you go to an extremely influential paper published by evolutionary biologist Ernst Mayr at Harvard. In 1961, he published a paper called Causes and Effects in Biology. And what I want to draw from that paper are two concepts that I'm going to come back as I discuss how we try to do causal inference: proximate causes and ultimate causes. Just think of those as, in proximate causes, we're trying to understand why things happen in a mechanistic way. It can be the cell, can be the organ, can be the individual. You can think of physiology, you can think of energetics, you can think of endocrinology. Ultimate causes, Ernst Mayr was developing this paper, from what I read, in trying to bring some balance to what was a very strong tendency to focus on genetics from the 20s and 30s. He himself, being an organismic evolutionary biology, thought that we also needed to understand causes at an adaptive level, functional level. And that's what it's been referred ever since as ultimate causes. Well, I'll come back as I examine what progress have we made in understanding proximate and ultimate causes for the phenomenon that are of my interest. At that time, this is the top panel of a figure of a paper that I'll show next. You can see that the understanding there was that causality was rather unidirectional. It was bottom up. You just go from the genome in a very programmed way to the cell, the organism, and the environment. And whatever that happens in that generation 1, that's pretty much inherited to generation 2. And that was set. And of course, this has changed over the years. And now for a long time, we've developed subdisciplines like Evo-Devo, where we understand that causality, we need to understand causality at different levels. Yes, there is an influence of the genome in what comes next, but so there is influences of what happens at the level of the cell, of the organism and the environment, and it is not unidirectional, but it is reciprocal. And some of these changes that occur during ontogeny, during your lifetime, may be also influencing the following generation. So we have moved away, most of us, I would like to think, from what all of you are familiar with, which was so influential as well, and with positive and negative consequences, as portrayed in the Selfish Gene book that Richard Dawkins wrote. It's almost 50 years ago. So that is what we call these days the extended evolutionary synthesis. How do we do this within biological anthropology? In a nutshell, biological anthropology, which we also call evolutionary anthropology, physical anthropology, we are trying to understand what it is, what it means to be human if we take biology and evolution into account. So when you study humans, of course, we have come to realize, embrace, include the component of culture. You cannot understand what it means to be humans if you don't consider culture. But something that it's a rather challenge these days to communicate in classroom is that I am of the firm opinion that everything begins with biology. Because at the end of the day, we are made of cells, we're made of genes, etc. But you cannot ignore culture either. So these are some questions that we ask, both on a very deep time scale and also a very broad spatial scale. What makes us different from other living organisms? For example, chimps. When did we really start cooperating for producing or obtaining the food, as the drawing on the top right shows you? And once we got that food, when did we start making fire? Were we using the fire to warm ourselves? Were we using the fire to cook? And now that we cook, suddenly our complete nutritional functions change. When did we start walking on two legs? These are some typical classical questions that we ask in human biology, in biological anthropology. And then we go on to get data. And we never get all the data we would like to get. This is, by the way, I was thinking, there's something I keep telling students all the time, and I think that you'll appreciate and agree with this. There is this, we forget that we are never getting the data we would like to get, right? We get what we can, not what we would like to. And the students don't understand it. And sometimes I feel like we forget that when we're reading papers or reviewing grants. The kind of evidence we can get, fossils, that is, I would say that's a rather unequivocal, we can hold a skull, right? We can hold a stone tool, but we can quibble about who made it. Caring and storing, we can look at material culture. Genetics, there is information on genetics that can help us reconstruct the past. Behavior, and that's my focus. The catchphrase for behavior is behavior doesn't fossilize. And one of you showed yesterday, I apologize, I think it was Thomas, the first slide was some footprints on a beach. And then we looked at a track of a fox. Well, what do we make of that? What do you make of that if now you find a footprint that you know has been dated to 1.3 million years ago in the savannas of Africa? You can make of that a lot. How have we used these approaches, and what are the kinds of questions that are closer to what I've been doing for this time? One of the things that so many of us wonder is, in what makes us humans, what makes us love, right? Well, what are the proximate and ultimate causes that have developed a living organism that can connect emotionally with another living organism in a very peculiar way that we don't see in many taxa, right? We refer to that establishing some pair bonds. What is it that makes some living primates develop this kind of emotional bond in a very peculiar way with their with young? I'm not going to call them their offspring for a while. We'll see. We'll come back to that. And some other living organisms don't do it. Paternal care. More specifically, we're narrowing down to the foci of my research. Why are some primates pair living and socially monogamous? Why are some males such excellent fathers? Those are really the two central questions that I want to use to share some findings, but probably even more than that, share some concerns, some limitations, really embracing the uncertainty that there is in all that I've done. Much of the research that I'm synthesizing in merely 17 minutes has done in collaboration with hundreds of students, assistants, and some colleagues. These I wanted to mention. Anthony Di Fiore, we were students together at UC Davis when we were grad students 10 years ago. Marcelo Rotundo, you didn't pick up that, you're not paying attention. Marcelo Rotundo has been the field manager in Argentina, working with me for 30 years. Maggie Corley and Alba are two junior colleagues who have joined the project now for some time. And again, going back to when I said, listen, we need to do something. So it was 2019, that we started working on this paper that we published in the Yearbook of Physical Anthropology, summarizing the research we've done with some of these primates. In fact, it took us 5 years. No, it was published in 2020. We started working on this in 2013. There was a science paper published by one of my colleagues, the most famous social evolutionary biologist, Tim Clattenbrock at Cambridge, making some claims about the evolution of monogamy. And let's say we did not agree with many of the conclusions, and we just do not have it in us to produce the quick turnaround to refute some of the claims. It took us 7 years. It's the kind of work that it's thorough, it's deep, doesn't make it into science. You cannot summarize it in a tweet, but we published a 60-page paper that we're very proud of because we really got into the details of the issues we had with the inferences made for the evolution of social monogamy in mammals. Our labs. We have labs in the forests of Ecuador, Peru, and Argentina. We've been working there for anywhere between a few years and 28 years in Argentina. And I work also in labs in the States, where some of my colleagues in Bastrop, Texas, and Davis, California, are responsible for captive colonies of some of these pure living taxa. I'm beginning to look back to my background, the importance of tackling the same questions from different experimental approaches, and the power of moving back and forth between what a lab can offer you and what the natural setting of the field can offer you, and drawing the best from each. I will not be talking about this, but we're going to have 15 minutes. If any of you, so those of you traveling from Europe who are still trying to figure out how to deal with jet lag, we've done a lot of research on the evolution of diurnality and nocturnality, because the owl monkeys that I study in Argentina are not nocturnal. We are not working during the night. I did that when I was young, not doing it again. It doesn't pay off, among other reasons. But just bring this up during the questions if you want to learn more about that. Tons of students, assistants, I want to thank them. We do that a lot. Please let your own students reach out. We always have opportunities to bring young scholars to the field to do research with us. And we have some wonderful facilities. We have a field station in the city of Formosa. We can host researchers, collaborators, and we have camp. Now we're going to Argentina, the lab in Argentina. And the bottom right image is showing you roughly 200 hectares, 500 acres, which is really our main backyard. That you can see in yellow, the system of trails. One of you was asking me, what do you do? Well, we produce the data that feeds into those documentaries that hopefully some of you have watched and enjoy. BBC, Netflix, Life on the Planet, David Attenborough. We're producing the data that goes into that. That's the kind of setting, that's the kind of lab where we are. I want to go more deep because I was thinking about it. When was the last time you took a course on evolutionary biology, or that you heard about evolutionary biology methods? My impression is that for most of you, it's been a while. So I felt it would be interesting to hear. I mean, I was learning from you what happens day in, day out. So I want to share a little bit of that. What happens day in, day out is that you go out in the forest. You wake up early, you drink mate, and you really have to go find the subjects from which you're going to be collecting data. And how you do that, it's a challenge in itself. More and more we are relying on some of the chips and devices that some of you have been telling us how you produce. In this case, what we're doing is we're capturing owl monkeys with a rifle with CO2 darts. Sounds easy, it's an extremely complicated procedure, and we're feeding them with telemetry collars to track them in the forest, or we're also feeding them with accelerometers, that your iPad tracker. We don't know how many steps they need to take to stay fit, but we can collect a lot of data that summarizes their activities. So we do that, and that allows us to find them. And when you find them, it depends on what you're doing, it depends on the day, it depends on the samples you're collecting. You may be collecting just direct observations. It's funny how we've been talking about observations, right? I mean, my goodness, could we spend a week discussing what we mean by that? Some of us are calling a footprint an observation of what? It's only of a footprint, since the fox wasn't there, nor the human was there, right? So sometimes we're literally collecting data from what the animal is doing. Sometimes we're collecting monkey poop from the ground. Sometimes we're just going through the forest collecting what we call life history data, get a sense of what's happening in each of those families. We've been doing that year-round. And this is, I mean, I need to go tell our dean that it doesn't stop. We need to keep doing it year-round. We need to be in the field. The monkeys don't stop. The soap opera continues. And we've been doing that non-stop since May 1996. When we do the maths, and it's not big data by any means of this workshop, but it comes down to imagine that for 28 years, you've had somebody in the forest 4 or 5 hours a day, every single day of 25 years. That captures the effort that has come. In terms of genetics, in terms of ultimate causes, I mean, can we start looking at what happens over an extended period of time? We're looking now at the 4th generation of identifying individuals. These are not fruit flies, but they're not chimps. So there's a lot of power in studying a system where you can be looking at great-grandchildren in 20 years, something you cannot do with whales or elephants. We collect genetic data. One of the reasons we capture them is because we can produce high-quality genetic material for some of these animals, of known sex, known date of birth. We have cell lines established as part of the collection that I developed for the San Diego Zoo. Sometimes we collect tissue, and other times we just collect feces. Again, methods. What does it mean? We are going to come back to that. I mean, what does it mean when I estimate some mediator of a potential causal path from something I measure in poop, in blood, in an individual cell? We collect behavior, life history, demographic data, and of course we have a very comprehensive panel of variables to say something about the environment. Why are some primates peer living and socially monogamous? By socially monogamous, I'm just saying that they spend time together, right? Without going into the details of what may or may not happen in our modern societies, people live as a pair in one apartment. God knows what happens when they're not there, but we call them socially monogamous. We're going to tackle that first question, and I want to go slowly on this one. So I want to capture your attention again, please, on purpose. I'm keeping the text. We're going to read slowly because it's central to understanding the challenge of the inferences we're trying to make. Let me just say, to give you a little bit of a time scale, is that these owl monkeys, if we were to connect them on a phylogenetic tree to humans, we probably need to go back 40 million years ago, if I remember that well. So we're taking a big leap in the inference. This is from a paper. It was a note. Actually, this is one of these editorials on the article that I was referring to before by one of my colleagues, Evolution of Mammalian Society. The hypothesis is the narrative model that we want to explore says that the distribution and abundance of food, top left panel illustrated with some fruits, or maybe the predators, the middle photo, or maybe the pathogens, the bottom, yeah? Those potential factors, it's unfortunate that it says here, determine, they are associated with whether females of a given mammalian species are solitary or gregarious. Now we move to the right. A female may be socially solitary or gregarious in relationship to food, to predators, to the risk of transmission disease. The hypothesis is that male social strategies are determined in a second step by infanticide risk. How much a female is concerned, there's a risk that some male may come and kill her infant. Options for parental care, engaging males in the care of a young. Intrasexual selection, by that we mean that it's within the same sex, males fighting between them for access to those females. All of that is going to determine the spatiotemporal distribution of receptive females. So we move to the right panel, and depending on these conditions, we're expecting, we're predicting that we're going to see a social arrangement along pairs, one male and one female. We may see solitary females with some males roving over the ranges of them. Or we may see one male holding control to several females. We may see several females and several males together. That's the idea. This drawing, in a number of forms, has profoundly influenced all research done in primatology and to a large extent in biological anthropology. Of course, we can make it a little bit more nuanced, and you can also draw this. How on earth do you go into our forest and try to produce data that will give you a sense of satisfaction? These days I worry about that more than whether it's going to get published or not, that you have really made progress. And you have no choice, at least I don't know of any other option but to choose a section of that. And I just want to illustrate what we've done. For that top right corner of the graph, we said, okay, let's collect some data, define some variables that relate to the distribution of males and females in space, to see how much they move, how that may contribute to understanding the ability of a male to monopolize access to a female. Right? The ability of males and females to mate with other partners. You're really imagining yourself, this is what I tell the students, you need to decide, can you have two girlfriends? Well, maybe if one is in New Haven, the other one is in Milford. Can you have two boyfriends? Well, maybe now with Zoom, if you decide that you're maintain those relationships virtually, but you cannot have girlfriend or boyfriend, 10 of them, in 10 different countries all over the world. And that's a little bit the exercise we're doing. So Owl monkeys, one of the 3 taxa that we study, everywhere you look at, always you find them pair living, what we call socially monogamous, and pair bonded, meaning that we infer that there is this preference. And that's, thinking about humans, that happens all over the world. Humans are developing a primarily relationship with one single other individual. It's changed, can be the same sex, different sex, same gender, but there's always a preference for one over the others. Owl monkeys, always pair living, always pair bonded. This is the forest I was showing you before. You go into our forest and you have 18 of these groups. And they're really distributed in space like I'm showing you there. Remember, that's roughly 200 hectares, 500 acres. Let's take 2 of those backyards, right? Think of them as backyards. They're roughly 5 hectares. That's maybe 12 acres. So these guys are holding a backyard of 5 acres, 1 male, 1 female. There's another group that has their neighbors, another 5 hectares. The problem is that we also look around, and in these neighborhoods, some of them got to really have twice as much area, but they still have only 1 male and 1 female. So what we ask ourselves is, why is it that if some of these males have the ability, the proximate causes, the mechanisms to really range over 10 hectares, why don't we see some evolution that produces a male that now is going to range over the ranges of these 2 smaller territories in red? Why don't we get a drive for the evolution of a polygynous male? If the guy can cover 10 hectares, if he can monitor 10 hectares, why don't just monitor 2 territories of 5 instead of 1 of 10? The answer is we have no clue why not. So that's really, after 28 years, there is no, it's very, very difficult to resolve this. What we're trying to do now is get into much more detail in measuring the distribution of resources, which is extremely challenging. Why, some of these males, actually all of these males, are wonderful fathers. Why are some males excellent fathers? And that's a fascinating question in evolutionary biology. Let me tell you why. You can connect with these guys being wonderful fathers, right? Male penguin keeps an eye on the chicks, mom goes fishing and finding food. Seahorses, you probably know that the males hold the pregnancy. But mammals are very, very different. For one thing, fertilization is inside the female tract, and that generates father uncertainty. And second, mom, as opposed to the penguin and the seahorse, mom is stuck with the investment, with the demand for energetics during pregnancy and lactation. So it is a puzzle in mammalian reproductive biology why males would commit to a single female in a socially monogamous relationship. One possibility, we do know that our monkeys provide an awful lot of care. If you have children, if you have grandchildren, encourage them to browse the web, to look at videos of our monkeys taking care of the babies. It's fascinating. A couple days after the baby is born, the father is primarily involved in taking care of the young. Except for nursing, the male does it everything. And we do feel, based on data from the lab as well, that the infant has a stronger attachment to the father than the mother. That's remarkable for a mammal. They carry the infant, they share food with the infant more than the mother, they groom, they touch, they play. They're the primary caregivers, of course, leaving aside nursing, which is not trivial. We wanted to understand what are some possible proximate causes of the involvement of the father with the care of the young, how that may be feeding into explaining pair bonding and monogamy. And again, on purpose, I just pasted the whole text of this article because it illustrates a little bit what I'm trying to get traction on in my own discipline. Notice the paper is from 2021. And for 3 years, a whole NSF grant went on to collecting monkey shit from the ground, hoping that, but this is the path we're trying to connect. So what is the hypothesis? The hypothesis is that for a male to engage with a female, one possibility is that the male is helping the female reduce the energetic burden. If I help you take care of the young, then you can rest, then you can sleep. So we want to look at endocrinology. We want to understand if cortisol, which don't think about cortisol anymore as a stress hormone. That's all, throw it away. It's much more than that, and we're thinking of it as a metabolic gatekeeper. Levels of cortisol are related to how we're metabolizing glucose, right? But that matters in the brain. I mean, we need to, if I could get the data I wanted, I would like to know what's happening with cortisol and cortisol receptors in the brain. I cannot do that. Well, can you do blood? No, I can do that. Can you do urine? No, I can do that. What can you do? Well, I can collect monkey poop from the ground and hope that as the monkey excreted or whatever it excreted, something in the poop gives me an adequate proxy of cortisol in the brain. We did that. This is an unusual note and section for our journal and our discipline, but we just wanted to do it. And we, more and more, we're doing multi-model selection, model averaging, but we can play with numbers all you want. What we cannot allow ourselves to forget is what is the quality, the internal validity, the robusticity of the variables that we have. And more, with my colleague and wife, Claudia Vallejo, we're feeling like, is it worth to put all this effort in trying to infer something about the energetics of parental care from a measure like the one we can get? Ultimate causation, what are the benefits of paternal care? Huge survival of infants. We think that may be part of the explanation of why we're saying this. You do not get these rates of survival in any other primate. For a wild animal, they are doing wonderfully, and maybe it's associated with having both parents provide care. The other ultimate question is, okay, if this is adaptive, if we can imagine this is going to evolve over time, the same way I was talking about, why don't we see a polygynous male strategy evolve? How come did we get to have a male care strategy to evolve? For that, we need to understand if there's father sonity, and if the males who are keeping an eye on the females are really the ones who are siring the young. It's crucial that we go from describing their pattern as sexual monogamy, social monogamy, they're hanging together, sexual monogamy, they are copulating with each other, genetic monogamy, they're really only producing young with each other. And we know now that the system we have, it's quite extreme because we have no other primate for which we have the kind of data we do. We have now tested 120 dyads of putative fathers and offspring, and not a single time we have assigned the paternity to another male that it is not the putative father. Most of you are familiar with the fact that when we assess paternity, we're not confirming the identity of the father, we're simply just deciding which ones are not assigned paternity. So that may be another answer, partial answer to the evolution of this, that a male who is committing to a monogamous relationship now succeeds in siring offspring that in turn probably have that tendency to provide care as well and stay pair living and pair bonded. So the idea is that maybe you really have pair bonding as a necessary, but we don't know if sufficient condition to then facilitate the evolution of fatherhood and paternal care. You can read about all this. I added this because the talk is going to be public, so you can see the Yale Today for a whole piece on this, and a book that we just edited only this year. It's bringing together 30 years of research on animal monkeys all over Latin America, and you can really find there some good synthesis of the research that I have presented. This is the last slide with the list. I mean, really, this is a shopping list of the things that I would like our discipline to consider some more. It is primarily observational. We normally have very few subjects. This is not for my discipline, but I noticed yesterday we didn't really talk, and I invite comments on this. We didn't really get in our conversations and presentations on what differences are we going to make when we're talking about prediction for the individual and prediction for the population. I think we have a lot to discuss there because the public, they're not getting it. They're confusing our talking about we can model, we can predict, but we're not telling them this is not going to help you. Sometimes we have no clue about what are important effect sizes that we want to find or report or document. We literally have no clue. I don't know what is the difference in cortisol as estimated from poop that really means something to the animal. There's no discussion in my discipline about the difference between noise and bias. Actually, I think it's not happening in any field, right? Now everything's a bias. Well, these are two different concepts. I recommend that book that Daniel Cunningham just published. It's quite interesting to get the basics of the distinction between those two. Who was it yesterday? Samantha made me think, oh my goodness, missing data. Do we have work to do there? So there are things we cannot do, but all of these we can. Let's do them. If you're missing data, think about them. Let me know why you didn't get those data. I mean, what are the causes of missing data? That's huge. We're not doing it enough, I think. For the students, they confuse data and inference from data all the time. And then all of this we summarized in a commentary to a journal of our main society that we need to keep working. This is central to the workshop on the difference between statistical inference and causal inference. Thanks to all the agencies, and I thought that would be a nice collage to finish.
Thanks so much, Eduardo, for opening eyes to very long causal chains that are needed in inference in your discipline. So we're open for questions. Julia, Jaco.
That was fascinating. So I learned that Barbary macaques are also excellent fathers, but the reason there is these groups live together, and because paternity is uncertain, the males look after all the children just in case they're theirs, and actually males form bonds in Barbary macaques over the looking after the young. So I mean, the result is the same, that they're very good fathers, but actually the relationship they have with the females and the males have with each other are very different. So I was wondering, even in different types of creatures with maybe similar type of habitat, similar type of resources and so on, you get to the same outcome from very different for the very different reasons. And how can you untangle all these things, and how does, more broadly, is it clear what determines whether a group lives together, and there's either a bigger group lives together as a patriarchy or matriarchy, or there's just pairs, basically, in this case?
So, thanks. I'm reminding myself that this is going to be recorded and made public, but I'm not going to say anything that I haven't already published in writing. So the Barbary macaques, there's one single paper, empirical paper from the 70s. It was a relatively short paper on Barbary macaques that again and again gets drawn into the same pool of all studies without distinguishing how robust the information is. My understanding is that Barbary macaques, some colleagues of mine have started to work on them again. But the outcomes are the same, but the extent to which the questions have been examined, I don't think they are. So going to but what matters, what you said is, I think it's interesting that it is true that it's difficult to measure. We have tons of papers discussing how we're going to define paternal care, right? I mean, think about your own families. What does it mean that a parent of yours is taking care of you? More and more we're learning with studies across societies or human societies that there could be a lot of observational learning that the young do from the father. But if we're looking at the father, I mean, if we didn't know that the son is there, we would not be describing this adult male as teaching anything. But the child is learning from the father just through observation. So we make, in a very simplistic way, we talk about direct care and indirect care. The Barbary macaques, like every single other primate I know except for the TD monkeys, do not have, in any way or manner, the kind of direct involvement with the young, transporting them, caring. I mean, the baby yells and that goes to its rescue. Now, it is possible, absolutely possible, but much harder to document, that the mere presence of adult males are providing some extent of care, because those males may be a barrier to potential predators. Those males may be a barrier to potential males who may kill those infants. So how we assess, then you brought up the word bond, which I did as well. And that's a rabbit hole because, again, how are we going to define bond? We do not have, I use the word for our research in the wild, but if you ask me, I mean, I will say here on record that I don't know if we have any solid data to speak about a bond for the owl monkeys in Argentina. This is another way in which in biological anthropology we can make our inferences more robust. We can draw, we can use the process of abduction. We need to do comparative biology because the way in which I can do it is, okay, I don't know if a male and female owl monkey, I don't know if the male and the infant have a bond in the sense that if I cut that relationship, there's going to be distress. I haven't shown that. I haven't shown that they will seek each other because they're always together. I cannot separate them, right? But I'm started looking around and I see there are all kinds of studies spanning decades on all kinds of other systems where when they see what I see, they have also shown that there is a substrate, that there are proximate mechanisms, there's a physiology, there's an endocrinology that speaks of a bond. We don't have that for barbary macaques. Technically speaking, we don't have it for owl monkeys. The closest we get is to some research on callitrichids and titty monkeys in captivity, where there is some wonderful work documenting the bonds. So how you get to that? Barbary macaques live in groups where you have several males and several females. And again, we do not have evidence that the relationship between males and females in those groups is similar to the relationship you have in pair-living taxa. But how we really make progress in understanding how we got there, it's profoundly difficult. I do have hopes in terms of the genetics, but the genetics then also seeing how the genetics unfold, right? We need to make so much more progress in the black box between genes or genomes and this very, very complex phenotype.
Yep.
Thank you. It's a wonderful topic, and I was wondering if you could say a little bit more about the statistical norm that was just mentioned in the paper that you wrote, and whether there have been discussions and people have been questioning this statistical norm. So in the paper, in the one more back is the there's a footnote in your third to last slide. Yeah, the p-value.
Oh,
yeah,
this one.
Yeah. Can you say a little bit more about that?
Yes, absolutely. Don't get me started.
Oh, but maybe you shouldn't say something. No.
I'd love to. I'd love to. I have 2 minutes. Already in the 90s, I was trained by psychologists. They knew about meta-analysis. There wasn't anything new about it, right? They knew about meta-analysis. But already back when I was a grad student, I realized that hypothesis significance testing was profoundly inadequate for everything I was doing. And I still had tables full of stars. It looks like a cosmology experiment. So now in 2021, again, through this period, we decided that we wanted to share with our colleagues, but this was because I was following with my colleagues at the American Statistical Association and the statement that the American Statistical Association published in 2016 saying misuse and use of p-values. They needed to include the use in the title, but the bottom line is 99% of people are misusing p-values and statistical significance, followed by the Nature paper with 100 signatories saying, let's stop doing this. And I'm begging you, stop doing it, because I think it's made so much damage for most research situations. Definitely, I'm speaking for ours. In biological anthropology, there are seldom any opportunities where you're going to make progress, definitely not in causal inference, by deciding whether this is worth reporting or not based on an arbitrary dichotomous cutoff, that it's the same no matter what you're studying. So again, I'll go on record. What was the reaction? That it was the first time in my life that the 2 reviewers said, this has to be published, and the editor decided to send it to a third. And the third said, this has to be published, and the editor, eventually, I guess, she published it. But why don't we see traction? I think it's because we need to train our younger colleagues to take responsibility. You are the expert. We need to go back to domain knowledge. I think it was Samantha again. The easiest is, no, I don't know what this means, but the P is 0.04. It ought to be important. That's what we're getting from students, a lot. And they're not thinking. I go back to my second slide. You have to know your discipline. You have to think hard. You have to think deep. You have to be open and modest with what you hope you can accomplish, and all that. It's absolutely counter to structuring your findings and publications around an arbitrary. For me, it's mind-blowing that we're still using 0.05, and we haven't. So there was a lot, but I'm optimistic. I'm optimistic. We're making progress.
Two quick questions.
Oh, one quick question, yeah. So, okay, sorry. So I have one. So the question is, what inferences do you have about how your monkeys choose their mates? Or to put it differently, what's the nature of love?
The nature of love.
Very quick answer.
Right,
to
the
this
nature
one.
of love, I
This will
suggest,
go
let's
on.
talk about it later. But the inference, how they choose, that's where you need a lab. That's where you can really, we have a lot of expertise in how we can assess preference. Be of mates, be of food, anything you choose. So we can do that in the lab. We haven't yet. Because the challenge is, even if we do it with a very carefully crafted study design, we'll still have relatively low number of subjects. And I suspect that the answers are going to be all over the place. It'll have to be done in the lab, and that's as much as I can say with the time we have. I'd love to, let's sit down, we can all contribute the same about the nature of love. I mean, I'm no expert there.
Yeah.
Thank you.
Yes, sorry.